Hi there. Today I am going to talk about feedback inhibition. Feedback inhibition or end product inhibition is a control mechanism that stops the cell from making more of the substance than it is needed and thereby wasting the chemical resources. In some metabolic reactions, several steps are required for the synthesis of a particular chemical compound called the end product, and this synthesis is similar to an assembly line with each step is catalyzed by a separate enzyme. In many anabolic pathways, the final product can allosterically inhibit the activity of one of the enzymes earlier in the pathway. This phenomena is known as feedback inhibition. Feedback inhibition generally acts on the first enzyme in the metabolic pathway. It is similar to shutting down an assembly line by stopping the first worker. Because the enzyme is inhibited, the product of the first enzymatic reaction in the pathway is not synthesized. Because that unsynthesized product will normally be the substrate for the second enzyme in the pathway, the second reaction stops immediately as well. Thus, even though only the first enzyme in the pathway is inhibited, the entire pathway shuts down and no new end product or even intermediate products are formed and accumulated. As the cell uses up the existing end product, the first enzyme's allosteric site more often remains unbound and the pathway then resumes the activity as before. The bacterium E. coli can be used to demonstrate feedback inhibition in the synthesis of the amino acid isoleucine, which is required for the cell's growth. In this metabolic pathway, the amino acid threonine is enzymatically converted to isoleucine in 5 steps. Step 1, Step 2, Step 3, Step 4 and Step 5. If isoleucine is added to the growth medium for E. coli, it inhibits the first enzyme in the pathway and the bacteria stops synthesizing isoleucine. This condition is maintained until the supply of isoleucine is depleted. When the supply of isoleucine is depleted, then the pathway again starts. This type of feedback inhibition is also involved in regulating the cell's production of other amino acids, vitamins, purines, and pyrimidines.